Well, good day. Thank you for the invitation to do this lecture. I'm really honored and humbled to be able to do this for this very important conference. My presentation is entitled Nitric Oxide, Revolutionized Lower Genitourinary Tract Therapies from Bench to Bedside. By way of disclosures briefly, there have been several financial relationships associated with research work in the field here that I'm presenting. So this talk centers on this fairly amazing molecular mediator, a gaseous molecule called nitric oxide, also known as NO. And for some, you may recall that the science in this field originated about 30 years ago. And Science magazine had presented it on the cover of a December 1992 issue, whereby it said, "Just say no, for nitric oxide," and proclaimed it molecule of the year. Its importance relates to its role in so many different areas in the biology of function. We know it has a role in immune function. We know it has had roles in hematology and cardiovascular research. And then it was described as a neurotransmitter involved in various neurological functions. And then perhaps what really was the clincher was it was studied to be the chemical involved in penis research. So as I present to you with this presentation, I have some educational objectives. First, to describe the role of nitric oxide signaling, the mechanism here, and some of the early discovery work, both in the penis, but more broadly even within the genitourinary system, the lower system there. But then also to review with you how this field now has been transformed for managing some disorders of the lower genitourinary system, and particularly erection disorders, including erectile dysfunction, and even moving forward the science of priapism, which is a prolonged erection disorder. So we're going to talk about all of this in this presentation. Now, I do want to give some acknowledgments right from the outset here and thank collaborators and mentors that have worked with me over the years. This work has come about across disciplines and many individuals. And this statement, I guess, harkens the African proverb saying that if you want to get there quickly, go alone. But if you want to go far, go together. And so my statement here is to say that I think we've gone far, and it has involved a number of individuals working collaboratively. I also want to acknowledge Professor Jeffrey Bernstein, who is a preeminent neuroscientist in the United Kingdom, who really humbled me when I received a personal letter from him back in the early 1990s, in which he congratulated me on some of the scientific work we were doing with this new gaseous molecule, nitric oxide. And this slide just makes the point that as he wrote the preface for a book called Nervous Control of the Urogenital System back in 1993, he did acknowledge that there are new neurotransmitter substances in autonomic nerves that have impact in the urogenital system. I guess the reference had to do with some of the nitric oxide work that was starting to emerge in the field, along with the various other kinds of scientific progress made in this arena. Now, as I begin to talk, I want to give you a reference to where this sort of all began. And for me, this was in the late 1980s, as I show you a nice schematic from the American Urological Association updates, which is a series of periodicals that teach lessons about where the field is evolving for erection, as well as a whole host of other urologic conditions. The one edition that came out on penile erection acknowledged that the neurologic system was critical for the erection response, mediating both psychogenic and reflexogenic mechanisms with neural circuits that would descend toward the penile area involving the autonomic pathways, sympathetic and parasympathetic. These are in balance, regulating penile erection, and then the notion that indeed, as it made sense that the neurologic system regulated penile erection, these were the essential neuronal supply and autonomic pathways involved. But what was not clear was what's the actual mechanism for penile erection in which the neurotransmission was executed at the penile tissue level. And many had worked in various areas of chemicals such as acetylcholine, various neuropeptides. None of these quite fulfilled the requirements. And so there was an ongoing research looking for the non-adrenergic, non-cholinergic neurotransmitter for penile erection. And that's kind of where the science of the field was about that time. For those less familiar, I'm going to show you a little bit more about the mechanism of penile erection. The physiology of penile erection is something that I think is a very amazing story to understand. The biology of penile erection is an amazing event in our bodies. And the two main corporal bodies, the corpora cavernosa, are situated on the upper side of the penis. The urethra runs beneath it, whereby urine exits the body. But these two main corporal bodies govern penile erection. And the structure is shown here with an artery that runs centrally with little offshoot arteries, and then the erectile tissue that allows blood to come in and fill the penis for erection. And shown at the bottom left side, flaccid state, on the right side, erection. The mechanism of penile erection has to do with how this tissue has to go from a contractile state and also with vessels that are vasoconstricted with flaccidity to that of erection, where these tissue expands, allow blood engorgement, the blood vessels vasodilate, and again, the tissue is in a relaxed mode. So this is the science of erection, but what's still elusive is what actually is the chemical pathway that regulates how this whole mechanism occurs. And that's about where we were at that time. Now, in the late 1980s, about the early 1990s is when the field really took a major step forward. And I salute various investigators in the field, including Lou Ignarro, Nobel laureate for his work in nitric oxide, along with some others working in this area. And then Jake Rafer, a urologist at UCLA, where Dr. Ignarro was working, got involved as well, looking at penile tissue. But let me tell you the story here. The story is that about that time, investigators were able to take penile tissue, initially in the rabbit, subsequently in the human, and do organ bath experiments where they were able to suspend the tissue in the bathing solution, and they could study the contractile effects of this setup. You can induce an electrical stimulus to make the tissue contract or relax. And if you made the tissue relax with a certain stimulus, the interesting observation of giving agents that are called inhibitors of nitric oxide synthesis, that you could then, with these agents, block the relaxation effect. And that allowed the scientist, Dr. Ignarro, to proclaim, wow, that means likely then that nitric oxide is the factor here that accounts for this tissue relaxation. Isn't that interesting? But at the same time, there was concern that nitric oxide is kind of a gaseous molecule, and how is it made physiologically in the body, and is it truly a neurotransmitter, and could it account for the neurotransmission required for penile erection to occur? About that same time, I was interested in the field, and this generated from some outside the box thinking about priapism. And this disorder, prolonged erections, occurs commonly in patients with sickle cell disease, a hematologic disorder, and they have prolonged erections, often in their teenage years and young adult years, which is pathologic and leads to damage of the penile tissue. And so I had the crazy thinking that maybe something was going on with nitric oxide and erection to some extent by work being done in sickle cell disease, hematology, nitric oxide studies, and other disciplines. And I was curious that maybe this is a chemical that made sense in the penile erection disorders to help us then understand the science of penile erection. Some of the initial work I did, shown up here on this side of the slide, is just using a nitric oxide synthase inhibitor, L-nitroarginine, with increasing dosages or dose response effect, looking at the erection response in the live rat now. So beyond the erectile tissue and organ baths, I was able to do live animal studies whereby I could induce erections with the neurostimulators in the pelvis of the rat that could bring about an erection effect, and then show that there's an intracavernosal pressure that could be induced with erection, and then using intracavernosal pressure and other parameters of the erection response, like tumescence rate, that with increasingly high doses of the nitric oxide synthase inhibitor given systemically to the rat, that we could block the erection effect. So this is a huge scientific physiologic advance then in a live animal, showing that this indeed was a physiologic mediator. But more than that now, teaming up with Saul Snyder, our mentor, and some other students working with him, his group had cloned the neuronal nitric oxide synthase gene, and then developed molecular probes looking at this in the brain and then describing it as a neurotransmitter in the brain. We were able to carry out some additional work in the periphery of the urogenital system and show that with these probes we can localize this in structures such as the penis urethra, the pelvic plexus, and other structures on a molecular localization level with immunoblocks. But then go a step further now, looking at actual cross sections of the rat penis. We're able to localize with immunohistochemistry that the neuronal NOS, nitric oxide synthase gene, is expressed in nerve endings as well as endothelium and even urothelium within the urethra, and that this is where these genes actually were active and could then be the source of nitric oxide, and accordingly fit the criteria of a neurotransmitter for penile erection. So this was a huge study that helped move forward the science of the field of penile erection. Very thankful to be involved with the scientific work, and this was published in Science earlier in the year of 1992, and with that, that contributed to the scientific research surrounding nitric oxide. We went on a step further now to confirm that the expression occurred in the human with studies of the nerve tissue in the pelvis and leading into the penis, similar to the rat studies. And so this substantiated the neurotransmitter claim for human penile erection as well. So the science of penile erection then really came forward in a major way in the early 1990s. And putting it all together nicely with this slide, this makes the point that there are a host of chemical factors involved: the autonomic nervous system, the balance between the cholinergic and adrenergic neurotransmitters, but also the non-adrenergic, non-cholinergic NAC mechanism, which nitric oxide is a major mediator, was significantly involved and accounted for the tissue relaxation within the penis that was necessary for the blood engorgement and the erection response. And a number of investigators have all contributed to this science, but nitric oxide certainly came to the forefront as a major mediator for mediating penile erection. Now, the research work in my laboratory over the next decade went further to try and clarify what the role of nitric oxide was in penile erection. One of the interesting stories was that as we obtained mice that were nitric oxide synthase knockout mice that lacked the neuronal NOS and endothelial NOS and even double NOS genes that were genetically engineered to have these genes deleted. We studied them for our sexual physiology research, as well as others looking at these mice for other biological functions. But here we were intrigued, as others were also challenging us, as to how do these mice preserve copulatory ability if they're lacking nitric oxide synthase? And that's something. So what we decided to do is explore this a little bit further, try to understand more about the science of penile erection relating to nitric oxide. One of the areas of research was that we actually were able to show that there are other variants of neuronal nitric oxide synthase, not just the predominant nNOS alpha form, but nNOS beta is also expressed and physiologically relevant with molecular probes specific for the knockout animal looking for nNOS beta. And we've suggested that there are redundant nitric oxide mechanisms that could account for penile erection. And this therefore explained how these animals preserved penile erectile function. Now, along the way, we also made some interesting observations with the endothelial NOS knockout animals. And there were two key observations we made. One was that with electrical stimulation of penile erection, these animals showed a supernormal erection response. They were just able to show very robust erections with minimal stimulation. And this gave us some insight into the mechanism of priapism, which I'm going to speak about here a little later in the talk. But the other observation was that these animals showed an attenuated erection response with pharmacologic stimulation. So what that is administering papaverine, which is a vasoactive drug that can reduce an erection response. Actually, we use this papaverine for men in the clinics who can inject this medication into the penis and create an erection effect. But we went back to study it in the animals, and these show erection tracings for the wild type, right here, the high line here, and also the nNOS knockout. But the eNOS knockout, endothelial NOS knockout animal, had a different kind of response. When we injected into the penis, they did not have such a robust response. It was kind of minimal. And of course, we measured the intracavernous pressure, the ICP, and with graphic representation here, we see that the eNOS knockout animals had a less robust response. So this was curious to us, and we said, this may suggest that there's something going on with blood flow and shear stress and how endothelial NOS is activated. And this helped us then move forward the science of penile erection, where we actually looked at mechanisms of phosphorylation, which activates endothelial NOS. And this seems to be an AKT-dependent mechanism. And this helped clarify that, yes, penile erection can be maintained through blood flow effects in the penis. And with all this abundant endothelium lining all of these structures within the penile tissue, this was a source of nitric oxide that was involved in sustaining penile erection. Now, we went even a step further with that, going back looking at neuronal nitric oxide synthase, because we still kind of were troubled that erection is more than a transient effect. It's more of a sustained biologic response. And that the neuronal nitric oxide source is really the predominant mechanism for nitric oxide release and then regulation. How can we make sense of that? And the science in the field was starting to characterize phosphorylation states of the NOS genes, as mentioned for eNOS. And there are activation sites on neuronal NOS as well. Phosphorylation sites, activated dephosphorylation sites, deactivating this enzyme. And we were able to show that there is a cyclic AMP dependent, almost a calcium sensitization mechanism, whereby this could lead to more sustained neuronal nitric oxide release of this gaseous molecule. And therefore, that was the additional mechanism contributing to penile erection. And we did some clever experiments whereby we measured phosphorylated nNOS with different degrees of electrical stimulation over time, showing that the expression level was there and increased. And then this would be a basis for thinking through that this is how nitric oxide is released more steadily in a tonic fashion. And then we did some nice physiologic work, and this is hard to see, but this just makes the point that compared to the wild type that had a great robust response to forskolin at higher dosages, measuring intracavernous pressure on the vertical axis here, that the other animals, including the nNOS knockout animals, were not quite so responsive to this forskolin dosing, forskolin being a cyclic AMP inducer. And therefore, we are able to show very nicely that the lack of nNOS interfered with the more robust erection effect of the neuronal NOS knockout animals. And therefore, we were able to put together the entire story here, which we then proceeded to publish, that integrated both neuronal NOS and endothelial NOS together, creating the erection effect. That neuronal NOS accounted for cyclogenic and reflexogenic mechanisms, both through calcium depolarization that is normally understood to mediate nerve stimuli and impulses, but also a calcium sensitization mechanism, which phosphorylates in NOS through protein kinase A phosphorylation. That also contributes to a more sustained nitric oxide release from nerves, along with blood flow that then by shear stress and flow across the endothelium contributed to the erection response, and this involved endothelial NOS with its phosphorylation by an AKT mechanism. So this now is where we put together the science of nitric oxide, the neuronal, the endothelial. This is how erections occur, and this currently now is accepted to be the science of nitric oxide mediated penile erection, and now in the textbooks of the physiology of erection at the molecular level. So my group was able to put all this together, and now we really have a good foundation for understanding nitric oxide mediating penile erection. Now, all of this is to say that we make scientific discoveries. We go a step further now to exploit this for clinical benefit. This nice graphic shows, again, the NANC, or non-adrenergic, non-cholinergic chemical nitric oxide for the brain, as well as other forms of stimulation, tactile such as that leads to nitric oxide release. And with its local release, it diffuses locally to the smooth muscle cell, the erectile tissue cell. And how this all works is that it drives the activation of guanylate cyclase that converts GTP to cyclic GMP. And this very potent second messenger molecule then accounts for downstream protein kinase G activation of smooth muscle relaxation and other pathways that account for penile erection. Now, the interesting science here is that we learned that phosphodiesterase type 5 that is highly expressed in penile tissue regulates this whole mechanism. And while there are companies starting to work at PDE5 or phosphodiesterase type 5 inhibitors for angina, heart blockages, coronary vessel issues, the patients in trials are having reports of better erections. And lo and behold, that's where the famous stories were that the companies took new interest to look at this for treating erectile dysfunction. But really, this had the foundation of the science of penile erection. And what we learned then is using a PDE5 inhibitor that can block the phosphodiesterase function to degrade cyclic GMP as a control mechanism. If this is inhibited, then more cyclic GMP can be released and induce the erection response. And so that's where the PDE5 inhibitors came in to be a transformative new therapy for erectile dysfunction. And answering the question of, is there such a thing as a potency pill? In 1998, Time magazine proclaimed a new pill had come out on the market, the Viagra, and along with its close cousins, these suffice as PDE5 inhibitors, and amazingly then did fulfill the notion of agent that could be a new therapy to treat erectile dysfunction. Now, our work extended beyond the penis, and early on we looked at other genitourinary tract structures. As I mentioned before, the urethra has a high expression level, lesser in some of the other structures. We studied this in the rat and then in the human. And then amazingly now, our focus did turn to the urinary outlet in the urethra and the prostate, whereby we were able to show nicely that nitric oxide function there is localized by expression studies in the nerve endings, less so in human benign prostatic hyperplasia. So therefore, there's a suggestion that men with lower urinary tract symptoms of BPH did not have as much nitric oxide in play, and maybe that accounted for their dysfunctions. We also did functional studies, and this mimics the work I explained earlier with organ bath work that we were able to do in collaboration. Dr. Takedo is a collaborator with this publication, and we were able to show in the human, dog, and others with the rabbit that the human prostate, the canine prostate, all showed a similar sort of erection, isolated tissue response, not erection, that is, the prostate tissue relaxation response following the nitric oxide pathway, that the various contractile effects could be influenced by using a nitric oxide synthase inhibitor that can reduce the contraction and reduce, bring about contraction because of lack of nitric oxide release. And then L-arginine being a source of nitric oxide, could induce more tissue relaxation, blocking the contractile effects. And this could be further shown, obviously, with sodium nitroprusside, which is also a nitric oxide donor. So all of this was meant to show that the physiology of the tissue contractile effects in the prostate also was nitric oxide mediated. And this lent support for some clinical observations that were starting to come about in time in recent years that have helped us understand that men having erectile dysfunction could have lower urinary tract symptoms, and those with lower urinary tract symptoms could have erectile dysfunction, and that there could be overlapping syndromes with coexistent pathophysiologic mechanisms related to nitric oxide. And this has been supported by other investigators who have looked at various other kinds of pathways, but certainly recognize that nitric oxide signaling could play a role in the pathophysiology of avoiding dysfunction and the urinary outlet. Now, my group has gone on further to look at sickle cell mice as a model of sickle cell disease in human beings. We've looked at the lower urinary tract, and just a brief statement here that we've looked at the nitric oxide, cyclic GMP, PD5 signaling pathway, not just as a pathway we're going to exploit just briefly, but that we were able to find and describe in this work is how we were able to modulate this system in a more sustained way and change PD5 function. And I just want to call your attention to the right side of the slide here, where we looked at nitrate treatment, oral nitrate treatment that had no effect in the wild type urethra with sodium nitroprusside. The relaxation response was really unchanged, but amazingly now the sickle cell mouse has a different kind of relaxation response, a little more exaggerated response at baseline. But when we gave nitrates, it normalized, resembling what you see in the wild type animal, suggesting that there is a regulatory mechanism of nitric oxide and PDE5, and we can actually regulate PDE5 function and alter the physiology. And perhaps this could be a way that we could move forward new therapies to help with avoiding dysfunction with chronic treatment. So this is an area of study that we also were very interested in beyond just the penis. But now I'm going to pivot slightly and get back to the priapism story, which is a near and dear area of scientific research. I certainly have seen many patients with sickle cell disease presenting in the emergency room with prolonged erections. This can also occur in other disease states. And while this sounds like, well, not so bad to have a prolonged erection, in point of fact, this can be a serious condition where a prolonged erection can lead to penile tissue damage. And so, as urologists, we have struggled to understand this condition. The science was unclear, but in recent years now, we've started to get some clarity on the subject matter. And fortunately, we may be able to develop new therapies here. My group has had a very major role in advancing this scientific understanding, and it goes back to the benchtop. And I mentioned earlier that the endothelial NOS knockouts, also known as NOS3 negative mice, have a robust erection response with electrical stimulation, and compared here to the wild type with an erection tracing with 6 volts, that gives you this kind of erection come on, and you turn off the stimulator, it comes back down. With the endothelial NOS knockout at 2 volt stimulation produces an equally robust effect. Wow. But the other interesting observation is when we turn off the stimulator, instead of the decline in erection being a little bit more immediate, it actually was prolonged. And the control of erection did not occur quite so immediately. So with that observation, we said, And I said, we got to understand this a little bit better. Let's go back and look at the molecular mediators of how erections are controlled. And lo and behold, PDE5 emerged in our understanding that compared to the wild type, the expression level is low. But what we also next did was said, well, can we show how likely this has occurred by reversing it? And we gave adenovirus constructs with NOS3 back to these mice, and we showed that their PDE5 expression came back up. And also their cyclic GMP production came back up. Isn't that interesting? So we were able to change the molecular biology, and the phenotype of these animals changed. They reverted back to a wild type phenotype. So their tracings looked more like the wild type, and they had more controlled erections when their PD5 function was restored. So we knew that PD5 function was key here in the mechanism of priapism, that the nitric oxide signaling had a role almost as a homeostatic regulator of how PD5 functioned. It was in balance with that. And oxidative stress may also play a role. But the main thing is PD5 is not static. It can be regulated, it can be modulated. It's downregulated in priapism. And the story here is looking at the normal physiology of erection as described before. Nitric oxide drives the cyclic GMP mechanism for inducing penile erection. In priapism on the right side, if the PDE5 function is intrinsically low, much like a PD5 inhibitor given transiently, and this has to do with nitric oxide being efficient, that the PD5 function also is downregulated, that then you don't have the control mechanism. You do not have the brakes that control how cyclic GMP continues to be functional, and therefore cyclic GMP is abundant and produces an exaggerated erection effect. And this is where we are able to make headway with this field and describe the science of priapism. And that priapism is a matter of unregulated cyclic GMP stimulation of the erection response. Now with that, we went one step further and said, let's exploit this here and see whether we can think about ways to treat priapism. And as I mentioned, PDE5 in the penis is regulated. And so one of the interesting stories was that we were able to show that if we can give nitric oxide back to animals that had this deficiency, and we did this in our sickle cell mice with a releasing compound, we could actually then control priapism. And so on the right side here, you can see a tracing of the normal erection response, the dotted line, compared to the exaggerated response in the wild type animal, that is in the sickle cell animal. And we can restore the wild type phenotype when we gave C6 prime to look more like the normal erection response with stimulation and then post-stimulation. And so this administration of NO releasing compound also we studied on a molecular level restored PDE5 expression levels from a lower level to a higher level once we gave this chronic dosing of the releasing compound. So PDE5 restored and the physiology reverted back. And this has led to some new scientific directions we're doing now, looking into therapies that can control priapism by restoring PDE5 function. And that's where our field is going forward now to deal with this erection disorder. So all of this is to say that nitric oxide is a very potent chemical in the penis, as well as the lower genitourinary system, that it has critical roles in regulating relaxation responses for penile erection in particular, has roles in erectile dysfunction, has roles in priapism, and as we have made scientific discovery, NO or nitric oxide is a matter of just saying no, and therefore has brought about scientific discovery clinically. And I want to thank all my collaborators for all the work that they've helped me with. I want to thank the organizers for allowing me to present this lecture that has reinforced our understanding of nitric oxide as the molecule of the year. For me, it may well be the molecule of a career, where I've spent much of my professional career in this space looking at nitric oxide and hopefully, to transform the field of urology. So I thank you very much for your attention, and I guess I'll sign off here. Be well. Thank you.